Hello. That's the best reception I ever got. So I'm going to talk really close to the mic because that's what they told me to do. But I have to tell you, as an infectious disease person, the first question I asked was, who else talked really close to this mic? So what you don't know is that when they ask scientists to talk, they have to send us minders. So I'm going to start out with my thank yous because I've watched the Oscars, and if you wait till the end, it never works. So I'd like to thank Allison and Ginger for helping me figure out what to and not to say. And then I'd also like to thank Hanley. Where are you, Hanley? Hanley, who's part cat, because you see her here in a box, right? So that's Hanley. She's my grad student who does most of the mosquito work that I'll be talking about. And also my student, Fausta. This is a picture of her right after I tortured her for an hour in her dental exam. So she was brain dead. She's in Rwanda right now. And then, of course, my two little monkeys, Evelyn, and Lionel. And this is pictures of us porch sitting on Sunday, right? Yeah, Sunday in Opelousas. So if anybody, who knows where Opelousas is? Besides, that's where all my people from. All right, so vocabulary. So when I first started in my program, I was introduced to this word arboviruses, and I was used to other words like the hilltop arboretum and arborist. And I was like, what do trees have to do with all these diseases? So it's not actually anything to do with trees. This is a naming fail. So what it is arthropod-borne viruses. So I have my cheat sheets here so you can see. So somebody got creative, and so we have arboviruses. And what these are is they're viruses that require an arthropod, like an insect, to transmit them. So the different kinds of arthropods that transmit are mosquitoes, which is what I work on, and then also ticks, and then we have sandflies, which are these fuzzy little things down here, and then, of course, culicoides or noceums, or biting midges. So all of these things have the potential to transmit viruses. They're not all here, so don't freak out. Let's see. Okay. So trivia. The two main mosquito species that I work on are Aedes albopictus and Aedes aegypti. So here on the left, this is Aedes aegypti. We don't have a lot of those in Louisiana. There are small pockets in New Orleans. There's a couple of pockets in Baton Rouge, but it's mostly in tropical areas. So when you hear about intense transmission of tropical diseases, that's the one that's responsible. Now, Aedes albopictus here on the, the left, the other side is the Asian tiger mosquito. So you might have heard about that mosquito. It's the one that if you're sitting in your backyard and this black and white mosquito comes and bites you, that's the one. So there are 3,500 about mosquito species, and there's over 156, I think, or 36, one or the other, Aedes species. So there's a lot of species of Aedes mosquitoes, but these are the main two that are responsible for transmitting the viruses that I study. So here's a caveat. This is about mosquitoes. I don't work on mosquitoes. I use mosquitoes to work on the viruses that I study. So we have an entomology department with a lot of cool people that can tell you way more about mosquitoes than you ever wanted to know. But I do know some trivia. So when mosquitoes take a blood meal, right, when they bite you, they're biting you because they use the protein in your blood to make eggs. So only females bite you, which means now you can use the correct curse word when you slap it, right? So you have been educated. So mosquitoes that are boys, right? They and mosquitoes that have already produced eggs, they are nectar feeders. So when they're not, aggravating you on your porch, they are pollinators. So they're not all bad, okay? So things you never thought you'd care about. Oh, by the way. How to tell the difference between a boy and a girl. So here on the top is a female mosquito, and Hanley took these pictures. These are really awesome. I'm going to turn this way. This is a female. She has, well, first of all, she's going to be bigger because there's no justice in the world. And this is her antenna, which is super not fuzzy. And then if you look over at the boys, not only are they constantly aggravating the girls, but they have these fuzzy antennae. So if you want to sit around and wait for mosquitoes to land near you and tell if they're a boy or girl, you can look at their antenna, or you can just wait for it to bite you, because if it's biting you, it is a There you go. So, let's see. Okay, moving on to the viruses. These are less interesting to make fun of because they're viruses. So I work on human health viruses like Zika, dengue, yellow fever, chikungunya, which doesn't have anything to do with chickens. Also, we work on animal health threats in these interesting named viruses, Bunyamwera, Batayan, and Gari, which is why I was in Africa a couple weeks ago. So these are all mosquito transmitted. They're mostly Aedes transmitted, usually Aedes aegypti, Aedes albopictus a little bit. But most of the transmission of these viruses are the little brown ones, Aedes aegypti. Let's see, okay. So some recent events. Dengue has been around like forever, and it was in Brazil, and they had very intense transmission. And then they had an Aedes aegypti eradication program in the mid-20th century, which was very successful because all the DEET. So then they found out that they stopped using DEET because it was having some ecological issues apparently, and they had a reestablishment of the Aedes aegypti population, and dengue reemerged with gusto. Since the 1970s, dengue has been there forever. It's there all year, every year, and it is a major public health threat. Dengue causes hemorrhagic fever, mostly in children, so that's why it's kind of a it plucks at the heartstrings, right? So there are 4 serotypes, and if you get the second dengue, the second time you get dengue is when you're at risk for hemorrhagic fever. West Nile. So West Nile emerged in the U.S. in 1998 to 2002. It spread across continental U.S., and now it's what we call endemic, and that just means it stays here all the time. So West Nile is primarily transmitted between a different kind of mosquito, a Culex mosquito, which are the little brown ones that bite you in the middle of the night, and birds. So when you get West Nile, you probably don't know you have it. If you do know you have it, you probably have encephalitis, and that's bad. But you don't get virus in your blood, and so therefore you don't actually transmit back to mosquitoes. The same things with horses. If horses get the West Nile virus, they also get neurological symptoms if they get symptoms at all, but they don't transmit back. So we are not a natural host for that virus. We are just what they call dead-end hosts. West Nile, if you are on social media, East Baton Rouge Mosquito Control, I have to plug them because they're actually one of the best mosquito abatement programs in the country. So holla. And they started doing a really nice summary of East Baton Rouge Parish collection and positives. So you can go on their website, or you can go on their social media on Facebook and Twitter and whatever, and see where West Nile is positive. And all that means is that you should wear bug spray, right? Okay. So of course, chikungunya came in 2013, 2014 into South America. There were some handful of cases in Florida. It came through and it swept through. There was over 1 million cases in a year. And that's primarily because nobody in the Western Hemisphere had really had chikungunya before. So there was no sort of immune pressure or no sort of like, everybody was available for the virus to infect. So it was like we had all this dry wood and a match. That's what happened. The same thing happened with Zika. So when Zika came in, it was the first time it had been in the Western Hemisphere, and so it came in, again, all the dry wood and a match. Now, that being said, Hanley has done a lot of work with mosquitoes, and I'll tell you how, with dengue, chick, and Zika is really, really good at infecting mosquitoes. So we'll talk about that in a minute. Yellow fever, there was a yellow fever outbreak in Angola, the country, not the prison, in 2013-14. And then there was also an ongoing yellow fever outbreak currently in Brazil. Now, there is a vaccine for yellow fever, but because of the Angola outbreak and demand now in Brazil, they are running low on the vaccine. And so you still have this sort of, they've had over 175 deaths just in Brazil in the last year. So yellow fever is still very much a problem. Okay, so what I study in particular is how fast the virus will emerge or get into a population and how fast it spreads. So we call that the transmission dynamics. That's what I study. So let's see. And how do we study that? We we use a combination of wet lab, so that's what we call wet lab, which is experimental, like at the lab bench, doing all these crazy molecular things, and also something, we do some field work, and then what we call a dry lab, which is mathematical modeling and statistics. So we have what we call a soggy lab, because it's both wet and dry. Right, so, ooh, talk close. So what affects the virus going from the mosquito to the person or the vertebrate, and then what affects the virus from going from the vertebrate or the person back to the mosquito, and then all the things in between. So again, we use, Evelyn loves math. She's my shy child. So how do we study this thing? So one of the things that we were really interested in is the time it takes. So the time it takes between a mosquito getting exposed to a virus, so taking a blood meal on a viremic person and then becoming infectious. So not every mosquito that bites a person with virus in their blood will become infected. And if they do become infected, not every mosquito that is infected will be able to transmit. There's a certain proportion of mosquitoes that are just going to like do their thing. We call that vector competence. So that is one of the main things that we study in my lab. And so what we have here is we have a female abdomen. She's been drinking some water over here because usually there's no gap between this layer of scales and that layer of scales. But here you can see we have blood fed this mosquito, and she is bright ruby red, which is good because it makes our job easier. We can see them. So we study vector competence. How do we do that? So, wait, wrong way. Oop, other way. Oops. So we used to, way back when I was little, walking uphill both ways to school, we had these glass things that you'd put blood in, and you'd have water running through it to keep it warm. And then the skies opened up, and angels gave us what's called a hematech, which is a 6-armed machine that you just get a little disc of blood. And so we order blood from a lab, because that's a thing people do. And we put virus in it, and we put like a really thin bit of membrane on it, and we put it on this arm that has a heater, and it keeps it at 37 degrees Celsius, which is approximately 98.6. And we put it on top of a cage of mosquitoes, and they just go to town. And it has made our life a lot easier. We get pretty good feeding rates on colony material, which is mosquitoes that have been trained to do this sort of thing. When we get mosquitoes from the field, it's variable depending on what they feel like that day. Albopictus, which are the Asian tiger mosquitoes, they're pretty easy to feed. So if you've ever sat outside and swatted at an albopictus or a black and white legged mosquito, like she then takes as a personal challenge to like, she's going to bite you, right? So she comes back and she tries to sneak around, and I find mosquito bites on my kids that I don't know how the mosquito got there, but they will get there. So we have pretty good feeding rates. We get virus into mosquitoes at pretty good rates. And then to tell if they have virus, what happens is a mosquito, let's see if I can go back. Oh, by the way. Okay. So turn this way. If you look at the mosquito's body, the legs are right here. So you see the legs. The salivary glands are about right here. So it's not like you think of people where your salivary glands are really close to where you spit. In a mosquito, the salivary glands are right here, or what we call the brisket. And it goes from here, and they have a little tube that goes all the way up into their proboscis, where they then, when they feed, or when they probe really, they spit. So what we do is we take the legs off of mosquitoes, with the tiniest tweezers in the world. Also, because it's cathartic. You get to go to, so what do? I pull the legs off of things. Like, what do? So we pull the legs off all these mosquitoes, and we test the legs. And if there's virus in the legs, that means they have a disseminated infection. That means the virus has gotten out of the midgut into the hemus cell, which is the mosquito version of a circulatory system. And it's not very complex. It kind of just goes all over. It goes through the legs, and then it goes into the salivary glands. So because the proximity of the legs to the salivary glands, we use that as a proxy to determine disseminated infections. And if they have a disseminated infection, then they are more likely to be able to transmit virus rather than just looking at infected whole mosquito. So that's how we measure vector competence. We use a lot of mosquitoes and a lot of tubes. By the way. So some other things have been recently with Zika. So the story with the mosquitoes is what we've determined is that Zika is real good at its job. So it's faster than most of the other viruses I've played with in Aedes aegypti. It gets to about 50% of exposed mosquitoes are infected at, 5 DPI? 6, 7? Look, she's like, I don't know. You are a fail. 5? Okay. 5 DPI. Dengue takes anywhere from 7 to 9. Chikungunya is like, I don't know, 6 to 7 days. So Zika's got very much an advantage over these other viruses when you look at them in a sort of, multi pathogen system. But some of the other stuff we did with Zika was, because we kind of got a jump on it, because we pay attention, we put it in some mice, and our pathologists, who are equally as morbid as we are, because they pull the legs off other things, they helped us look at the old strain of Zika that's been in Uganda since 1947, and probably before then, versus the new strain. So when Zika emerged in South America, it's really the first time we ever associated it with neurological symptoms, Guillain-Barré, which is a acute paralysis, and then microcephaly, which of course is the malformation of baby heads, and then retina scarring in children and infected adults. That was the first time we really had a lot of evidence that Zika was associated with these problems. So one of the questions we wanted to ask was, is this something new? And so because Zika was less characterized, we took the old strain, the Ugandan strain, and we put it in some mice, because apparently they don't let you put it in people. I don't know. So so one of the hallmarks of Zika that we've known about is conjunctivitis. So what we wanted to do is make sure that our model would recapitulate that. And indeed, we did find that these mice, you can't see, but they have crestulal eyes, and then they had, this is the pathology, they had cells where they shouldn't have cells. So this white space right here is supposed to be completely white, and all those purple things are cells that shouldn't be there. And what that shows is inflammation. So they had pink eye, right? So we knew that this was showing traditional symptoms of Zika, but what we also found was we did find it in the brain. So we could stain for the virus. We stained for the virus. I don't know if it's showing up, but, oh yeah. So these maroon or brown spots are where we have stained, the stain has taken for the virus. And we show that in the hippocampus, which is a part of your brain, as well as the cerebral cortex, which is a part of the brain. These are negative controls that makes us know that this is real. So we found that the old strain actually does infect the brain like the new strain. We found a whole bunch of other stuff, but the take-home message was the virus hasn't really changed. And so why then are we just seeing it? Well, my personal opinion, and since I'm the one with the microphone, is this is the largest outbreak of Zika that we've ever had on record. So it's a numbers game. There's so many people with Zika that even if it's a small percentage of people with these symptoms, we can pick it up. Just because, 1% of 100 is not that much, but 1% of 2 million is noticeable. Let's see. Field work. So sometimes I go get mosquitoes from around town. Now, when I was little in Opelousas, my grandma's house butted up against the St. Landry Cathedral graveyard. So me and my brother spent my entire childhood in the graveyard. So when I came home and I was like, oh, I went and got mosquitoes today, my mom was like, oh, where'd you go get mosquitoes? I'm in the graveyard. She's like, oh, it all makes sense now. So Aedes mosquitoes love graveyard vases. I don't know. So they also love containers. So when they say dump out containers because they have water in it to keep mosquitoes down, it's because these mosquitoes will preferentially lay their eggs in small containers like graveyard vases, toys, buckets, dump trucks, I'll tell you, are great, Lionel Joseph. So I get all my mosquitoes from like Tonka trucks. So this is us in New Orleans getting Aedes aegypti because that's where we can find them. And this is us in, if anybody likes jazz, anybody? Yeah. Bill Evans, jazz pianist, is buried in Roselawn. Is it Roselawn or Rosedale? Roselawn? Roselawn. You can go look it up. So this is the Bill Evans colony, but they died, but I'm going to go get some more. So if you want to go see Bill Evans' grave, he's totally there. It's kind of cool. Very nondescript. It's really weird. But anyway, they have a lot of Aedes albopictus there. You can get all the albopictus you want for free. They didn't even charge me. It's amazing. So it turns out Evelyn is a mosquito magnet, so I bring her along. Her dad was super thrilled. Oh, you find other things like frogs and teeth. That was interesting. But only the bottom, not the top. There were teeth in there. So the other field work we've been doing, like she said, I just got back from Uganda and Rwanda, looking at urban transmissions. So Aedes mosquitoes like urban areas because there's more places for them to breed, more containers. They'll breed in litter. So if you think about a water bottle cap, it's about that big, you can put 100 larvae in there and you'll get 100 mosquitoes out. So they love urban. But your idea of urban is Baton Rouge, New Orleans. Their idea of urban is Iquitos, Peru, which is a little more like what you think, right here. They like to breed in the house in Iquitos because it's cooler. But they also will breed in this kind of urban in Uganda and this kind of urban, which I don't actually know where we were at this point, somewhere in Rwanda. So urban is not always what you think. It's not U.S. urban. It's a collection of settlements or people's houses. It doesn't necessarily need to be that big that has a lot of stuff where the mosquitoes. Because, and what prompted the name of this talk, Aedes mosquitoes do not fly far. They will live happily on your porch, in your house, and never have to leave again because everything they need is there. Water and people. And sometimes they fly outside and get nectar if they need that. So when we're talking about transmission of viruses, mosquitoes are not the ones that move the virus. So when we're going in the field, we're looking at human traffic as well, or animal traffic, depending on the virus. Because my mosquito in this village 2 miles away is not going to transmit the virus to that village 2 miles away. It's going to be the infected person who goes from this village to that village and infects their group of mosquitoes. So with the Aedes transmitted viruses, human movement is also a very big component. Again, going the wrong way. So we're also looking at cattle diseases in Rwanda. We're looking at these because cattle and goats, like everybody has a cow or a goat. And so we're looking at how cows and goats associated with people increases risk of infection because, again, it's going to be the people and the cattle and the goats that are moving the viruses and not the mosquitoes. So that's some of the field work we're doing in Rwanda. So now's the part Evan loves, math. So when we're looking at human movement, what we're looking at is a person is either susceptible, which means they're available for infection, they're exposed, which means they have the virus but they're not sick yet, or they're infectious, which means they're actively got virus in the blood and they're available to transmit, or they're recovered. So one of the things we've been doing is looking at how human movement and being in your infectious period might affect the way that you're what we call the index case, which is patient 0. You're the person who went from this village to this village and sparked that village's outbreak. So without a whole lot of math, we basically took this one box of this one. So these are defined by time. So this eye is, let's say, 5 days. You have 5 days of viremia, which is when you have virus in your blood. And what we did is we expanded it to 10 days, and we let it vary. And the take-home message is, because I don't know, boo. take-home message is what our math was better than their math. So that's what you need to know. So all of this sort of feeds into a new old concept. It's an old concept, but somebody created a buzzword for it called One Health. And I think the vet school is a really good place that sort of embodies this. And zoonoses are animal infections that jump into people. So if everybody remembers the swine flu of 2009, that's a zoonosis. It was originally a flu that infected pigs, but it jumped into people, and it sparked this big pandemic public health problem. So the estimate is that 70% of emerging infections are either vector-borne, which means arthropod-borne, mostly, or zoonotic, which means they jump from animals to people. And that is why we really go into this sort of One Health idea, which is that human health, environmental health, and animal health are all related. And you can't have one healthy without the other two healthy. And so so what we're doing is we're taking a look at how environmental parameters such as temperature, urbanization, overdevelopment without considering these sort of public health issues might affect either animal and public or human health. So that's some of what we're doing. And I think with that, I will take questions.